Next topic is restriction endonuclease. Yes, restriction endonuclease cut palindrome. Yes, what is palindrome? REFER. This is palindrome. No. Many times in your books you will find few books they have given refer. It is palindrome. If you read in this direction or this direction, sequence is same. Palindrome means if you read in this direction or this direction, sequence is same, that thing is same, but this is not palindrome, because first thing, palindrome is double stranded DNA. Remember this point. Palindrome is double stranded DNA, just like GGCC. This is 5 and this is 3 and this is 5 and this is 3 and. So means double stranded DNA, if you read from 5 to 3 direction or if you write from 5 to 3 direction, sequence should be same. Palindrome means double stranded DNA in which if you write, you can write like this, you write both strand in 5 to 3 direction, sequence should be same. Yes, in double stranded DNA, with double stranded DNA in which if you write both strand in 5 to 3 direction, yes, alternative splicing occur naturally. Yes, means in our body, yes, one gene can give rise to more than one protein. That can be done by alternative splicing or RNA editing. Okay, this can be done by alternative splicing or RNA editing. So one gene one protein theory is discarded. We found that in our body the number of genes are decreasing. The number of genes are decreasing. Previously we started saying we excreted all the protein of the cell. We counted there were 1 lakh type of protein. Yes, we started saying that if 1 lakh proteins are there, there should be 1 lakh gene. But later on we found that one gene can give rise to more than one protein. So that's why number of genes are decreasing. Yes, now we see the number of genes are 20 to 25,000. Okay, clear? So yes, so this is restriction endonuclease in which if you write, palindrome is a double stranded DNA in which if you write in both direction, both strand in 5 to 3 directions, sequence should be same. Means suppose a double stranded DNA is there, the lower strand, if I write the lower strand in 5 to 3 direction, 5 G G C C 3 and this is palindrome. This is sequence is same, this is palindrome. Understood? So now you can say this is double stranded DNA. In this the lower strand, if you write the lower strand in 5 to 3 direction, the sequence is same, the upper. So in this question what is the answer? I have already told you. Yes, tag 1 restriction endonuclease commonly used to cut human DNA into pieces before inserting into plasmid. Which of the following is most likely to be the recognition sequence of this enzyme? Yes, GGCC. Note D. Yes, if you remember REFER, you will find wrong answer. This is not palindrome. So restriction endonuclease cut palindrome reason. Okay, so you can easily rule out other option. AAGG, CCTT. So here this is 5 and this is 3 and this is 5 and this is 3 and two strands are anti-parallel. First is A, first is C, so this can't be palindrome. In this A with T, first is A, first is T, it can't be palindrome. Yes, this is palindrome. This TAAT, if you see this TAAT, then you will say T with A, first is T, first is A, it can't be palindrome. Clear? Got the point? So this is restriction endonuclease. What is the concept of restriction endonuclease? Why they are called restriction endonuclease? We found that this is bacteria. In bacteria, there is restriction endonuclease. This can cut palindrome region. We found that suppose bacteriophage entered the bacteria. This restriction endonuclease will digest bacterial DNA. If it is DNA, RNA will be converted to DNA then synthesize more virus, so it will cut the DNA. That's why it will destroy bacteriophage. So this was protective mechanism of bacteria, that it prevent the infection from bacteriophage. Now what happened, that this bacteria was also having genome. So what is the protective mechanism? This bacteria has its own genome. So restriction endonuclease can cut the own genome of bacteria. So we found that yes, restriction endonuclease can cut this genome of bacteria. So we found that restriction endonuclease is also associated with a enzyme activity, methylation. So what happens, that this enzyme do methylation of bacterial DNA. Methylation of bacterial DNA. Methylation of bacterial DNA. Yes, methylation of bacterial DNA means it will, yes, when methyl group is there, then this restriction endonuclease will not cut this bacterial DNA due to this methylation. So methylation is a protective mechanism for cutting of restriction, means cutting of bacterial DNA by restriction endonuclease. So restriction endonuclease and methylation property are usually present in these enzyme together. Yes, AATT. AATT. Yes, it can be palindrome. Yes, it is palindrome. Yes, TAC1, TAC polymerase, Thermophilus aquaticus. Thermophilus aquaticus type 1. Okay. Yes, TAC. Bacteria are named like this. TAC means Thermophilus aquaticus. I will discuss with PCR. So this is clear to you. Now next point is euchromatin and heterochromatin. Euchromatin and heterochromatin. I should be a little bit more precise while teaching you, because if few of you are not understanding, that definitely I will try to give in a more precise way. Further you will not have this problem. Okay, don't worry. Methylation of bacterial DNA. Yes, I will take. Yes, methylation of bacterial DNA. What happens, that we know that bacteria, its own DNA is having palindrome. This restriction endonuclease is present in bacteria. Yes, this enzyme is present in this bacteria, restriction endonuclease. If bacteriophage will enter the bacteria, restriction endonuclease will digest its DNA. Okay, but this restriction endonuclease can also digest the DNA of cell, bacterial cell. So what nature did? Nature did the property of methylation, means this restriction nuclease also have a component that is methylation. That means the DNA palindrome which is cut by restriction nuclease, that portion of DNA is methylated. When it is methylated, then restriction nuclease will not cut its own DNA, but it will be able to cut the bacteriophage.